Hormones influence target cells by binding to specific receptors. The hormone-receptor interaction initiates the process of transforming the chemical message into the response of the cell. Your goals for learning are to learn about hormone receptors, to describe common second messenger systems and transcription factors, to review insulin, to understand that hormones induce changes in cellular metabolism. Here's what you need to know: that changing the shape of a molecule can activate or inactivate it. The terminology of carbohydrate, lipid, and protein metabolism. The difference between absorptive and post-absorptive states of the body. To see definitions of terms, click the bold red words. A target cell responds to a hormone with a sequence of events. Drag the hormone to the receptor to observe the events and response of the target cell. Receptor activation by the input signal, the hormone. Conversion of the input signal to a biochemical change in the cell. Conversions occur through second messengers and gene expression. Response of the cell. Examples of cellular response include contraction, secretion, transport, and synthesis or breakdown of storage molecules. We will study receptors here and conversion mechanisms and cellular responses later in this topic. Click Continue to proceed. The water-soluble hormones, peptides, and catecholamines bind to plasma membrane receptors on the outside surface of the cell. There are several categories of plasma membrane receptors, including receptors that activate G proteins and receptors with protein kinase activity. We will examine the most common G protein mechanisms and the protein kinases called tyrosine kinases that phosphorylate tyrosine. We will look at each kind of receptor, beginning with the receptors that activate G proteins. In the inactivated state, guanosine diphosphate, GDP, is bound to the G protein. Drag the hormone to the binding site on the receptor to observe its action. When a hormone binds to the receptor changes shape and activates the G protein. It releases GDP and binds guanosine triphosphate, GTP, a high-energy molecule. Part of the activated G protein travels along the membrane and activates an enzyme called adenylate cyclase. Adenylate cyclase catalyzes the conversion of adenosine triphosphate, ATP, to cyclic adenosine monophosphate. Cyclic AMP is a second messenger. The hormone is the first messenger. Cyclic AMP activates the intracellular enzyme protein kinase A, which can phosphorylate many other proteins, activating some and inhibiting others. This mechanism can induce many different effects within a single cell and different actions in different cell types. This second messenger system can also phosphorylate ion channels and alter their activity. The response of the cell ends when cyclic AMP is degraded to AMP in a reaction catalyzed by the enzyme phosphodiesterase. While active, G proteins keep stoking the pathway to phosphorylation of cellular proteins so that huge numbers of them are produced. This phenomenon, called amplification, greatly increases the effect that a single molecule of a hormone can have on a cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. G proteins, like the one we just described, that stimulate production of cyclic AMP are known as stimulatory G proteins. Inhibitory G proteins inhibit production of cyclic AMP and the subsequent response of the cell. Another type of G protein activates an intracellular messenger system that uses diacylglycerol and inositol trisphosphate as second messengers. Drag the hormone to the binding site on the receptor to observe its activity. When a hormone binds to the receptor The receptor changes shape and activates the G protein. It releases GDP and binds GTP, a high-energy molecule. Part of the activated G protein travels along the membrane and activates an enzyme called phospholipase C. Phospholipase C causes a membrane phospholipid called phosphatidylinositol bisphosphate to cleave into diacylglycerol and inositol trisphosphate. Each functions as a second messenger. Diacylglycerol remains in the membrane where it activates protein kinase C and inositol trisphosphate enters the cytosol. The activated protein kinase C phosphorylates other proteins, causing responses of the cell. Inositol trisphosphate prompts the endoplasmic reticulum to release calcium ions. Calcium ions also behave like second messengers. They can act directly on proteins or bind to the protein calmodulin that can act on proteins to cause the response of the cell. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. Drag each hormone to a binding site on the receptor to observe its activity. When a hormone binds to a tyrosine kinase receptor, the receptor joins with a neighboring receptor and changes shape. Each receptor phosphorylates tyrosine groups in its neighbor. The process is called autophosphorylation because the receptors are of the same type. The phosphorylated tyrosine groups provide docking sites where relay proteins bind. They initiate a series of protein phosphorylations that lead to the response of the cell. Receptors with tyrosine kinase activity often bind hormones like insulin and other growth factors that affect cell growth and differentiation. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. On this page, we will study mechanisms used by epinephrine and insulin to induce cellular responses. Nearly all body cells express receptors for epinephrine and norepinephrine, called adrenergic receptors. There are two families of adrenergic receptors, alpha and beta, with each member of the family designated by a number. All adrenergic receptors bind both epinephrine and norepinephrine, although with different affinities. All adrenergic receptors activate G proteins. Beta receptors are coupled to adenylate cyclase by stimulatory G proteins that increase cyclic AMP activity. Alpha-2 receptors are coupled to adenylate cyclase by inhibitory G proteins that decrease cyclic AMP activity, often blocking the increases induced by other agents. Alpha-1 receptors act via the IP3-DAG and calcium second messenger system. Some cellular responses to these catecholamines are listed beneath the appropriate receptors. Blood flow is redirected to working muscles where increased cardiac output delivers increased fuel and oxygen. Fuel molecules are released from stores and made available for use by working muscles. The airways dilate to maximize gas exchange. Note that all of these effects support the fight or flight response. Insulin acts primarily on liver, adipose tissue, skeletal muscle, and cardiac muscle by binding to tyrosine kinase receptors. Each tissue responds differently, and each has multiple responses. Some of the cellular responses to insulin are listed beneath the receptors. Glucose uptake increases in all three major tissue types. Insulin stimulates glycogen synthesis in muscle and liver cells and triglyceride synthesis in adipose and liver cells. Amino acid uptake and protein synthesis are stimulated in muscle. Note that these effects support the overall goal of promoting storage of fuel molecules. To review what you have already learned about insulin and to expand your knowledge and understanding of it, click an image. Insulin controls metabolic activity in the feasting or absorptive state that occurs during and immediately following a meal. The rise in plasma glucose levels stimulate pancreatic beta cells to secrete insulin. Rising blood levels of other nutrients, like amino acids, also stimulate insulin secretion. When blood glucose levels fall below normal in the fasting or post-absorptive state that occurs well after a meal, insulin secretion is inhibited. Hormones of the GI tract, particularly glucose-dependent insulinotropic peptide or GIP, and parasympathetic nerves also stimulate secretion of insulin. Negative feedback from insulin to the beta cells keeps the supply of glucose to the tissues relatively constant. This negative feedback system can be overruled by action of hormones or the autonomic nervous system, for example, when sympathetic nerves inhibit secretion of insulin. Remember that insulin is water-soluble and is transported in the bloodstream as dissolved particles. Dissolved particles are readily broken down, therefore insulin has a short half-life. The water-soluble insulin molecules bind to tyrosine kinase receptors on the cell surface. Once activated, the insulin receptor complex phosphorylates many proteins that alter many metabolic pathways. Insulin is synthesized in the pancreas in islet beta cells. The initial large pre-prohormone produced in the rough ER is cleaved into a prohormone and shuttled to the Golgi apparatus for processing. The prohormone is packaged into storage vesicles where it is converted to the active hormone insulin. Vesicles amass quantities of insulin for rapid release. Insulin is the major controller of fuel metabolism and is also essential for growth. We will examine the metabolic effects of insulin first, dividing them into effects on carbohydrates, protein, and lipids, then examine the growth effects. Insulin facilitates glucose transport into most cells by stimulating the insertion of insulin-dependent glucose transporters into cell membranes. Brain and liver are freely permeable to glucose at all times through transporters that do not depend on insulin. Insulin stimulates glycogen production and inhibits glycogen breakdown in liver and skeletal muscle. Insulin inhibits synthesis of glucose from amino acids and glycerol in the liver. All of these actions decrease plasma glucose levels. Insulin facilitates transport of amino acids into muscle and other tissues. Insulin stimulates protein synthesis and inhibits protein degradation. Failure to inhibit protein degradation is responsible in part for weight loss and protein wasting associated with untreated diabetes mellitus. Under the influence of insulin, the increased glucose in adipose cells is converted to fatty acids and glycerol, the molecules necessary for synthesizing triglycerides. Insulin promotes the synthesis of triglycerides and inhibits their breakdown. Triglycerides are storage molecules for both fat and glucose. To understand the control of metabolic processes during both the absorptive state and the post-absorptive state, we need to examine the functions of another hormone, glucagon, an insulin antagonist. Glucagon drives the catabolic activity of the post-absorptive period in response to declining plasma glucose. Glucagon stimulates glycogen breakdown and inhibits its production and stimulates glucose synthesis in the liver. All of these actions increase plasma glucose levels. Glucagon also promotes the breakdown of triglycerides and the degradation of proteins. I promote storage of nutrients. I make nutrients available. I decrease plasma glucose levels. I increase plasma glucose levels. I Anything you can do, I do the opposite. A second major function of insulin is its effect on growth. Insulin is the growth-promoting hormone during fetal development and is required for normal postnatal growth. Its exact role in postnatal growth is not fully known, but inhibiting protein degradation is of great importance. It is also required for the production of insulin-like growth factors, or IGFs. In summary, insulin affects both metabolism and growth. It shifts the body to anabolic metabolism during the absorptive state. Insulin stimulates transport of nutrient molecules into cells and production of fuel storage molecules and protein synthesis. These anabolic processes ensure that there will be a supply of nutrients for the post-absorptive period. Insulin is the growth-promoting hormone during fetal development and is required for normal postnatal growth. Bound by receptors, insulin enters cells by receptor-mediated endocytosis. Once inside, insulin is broken down by an enzyme system present in liver, muscle, kidney, and other tissues. Insulin receptors can be recycled to the plasma membrane. This occurrence downregulates insulin receptors and could account for the decreased insulin sensitivity of some cells to chronically high insulin levels. One way that scientists learn about the endocrine system is to observe the symptoms of patients who suffer from diseases that alter hormone secretion. Diabetes mellitus, the most common disorder of the endocrine system, results from a failure of insulin to direct the metabolic effects of the absorptive state. Click the liver to continue. There are two types of diabetes mellitus, often simply called diabetes. Type 1 diabetes, previously known as juvenile onset or insulin-dependent diabetes mellitus, is characterized by insulin deficiency or absence. Type 2 diabetes, previously known as adult onset or non-insulin dependent diabetes mellitus, is characterized by normal or elevated insulin levels but resistance of target cells to the actions of insulin. In the absence of insulin, or when target cells are resistant to the effect of insulin, glucose cannot be transported into cells. Recall the metabolic functions associated with insulin and answer questions to complete a flowchart that describes the signs, symptoms, and effects of diabetes. Click the word insulin to begin. Each question includes 2 bold words or phrases. Click the bold words that answer the question correctly. When insulin is deficient, will glucose uptake in liver, adipose cells, and muscle increase or decrease? When insulin is deficient, will the liver synthesize glycogen or break it down? When insulin is deficient, will synthesis of glucose increase or decrease? When cells do not take up glucose from the blood, and the liver produces glucose from glycogen and synthesizes glucose, will plasma glucose levels increase or decrease? Hyperglycemia is a hallmark of diabetes. In hyperglycemia, when plasma is filtered into the kidney tubules, the concentration of glucose is so great that glucose transporters become saturated and cannot remove all the glucose from the filtrate. Which of the following will result? No glucose in the urine, or glucose appears in the urine? Does the presence of glucose in the urine affect the amount of urine that is produced? Glucose induces an osmotic diuresis. Does the patient have to urinate more frequently or less frequently? If a person loses excessive water because of the presence of glucose in the urine, will they remain in water balance or become dehydrated? Do dehydration and thirst cause increased or decreased consumption of water? Despite the high concentration of glucose in the blood, cells are starved for fuel. As a result, will the patient eat more or less food? When insulin is deficient, will there be net synthesis of protein or net breakdown? Protein degradation in muscle causes muscle wasting. Will body weight remain the same or decrease as a result? When insulin is deficient, will there be net synthesis of triglycerides or net breakdown? If triglycerides are broken down, will the concentration of lipids in the blood increase or decrease? When lipids are used as fuel, they produce ketones, some of which are organic acids. The presence of ketones in the blood is called ketosis. When the ketones are filtered into the kidney tubules, will their concentration increase or decrease in the urine? Hyperglycemia causes two of the presenting symptoms of diabetes, polyuria and polydipsia. The third presenting symptom is polyphagia. Despite consuming large quantities of food, patients can lose muscle mass and weight. Diabetes can be treated. The treatment depends on whether insulin is absent or target cells have become insensitive to it. Click Continue to proceed. There are also several conditions that cause hypersecretion of insulin. Click the muscle to continue. Pancreatic beta cell tumors, or the overreaction of beta cells to glucose, called reactive hypoglycemia, cause hypersecretion of insulin. Too much insulin causes excessive transport of plasma glucose into insulin-dependent target tissues, resulting in abnormally low levels of plasma glucose or hypoglycemia. Recall that the brain is not an insulin-dependent organ. As glucose is transported into other tissues, little is left to fuel the brain. The symptoms of hypoglycemia, fatigue, sleepiness, the inability to concentrate, and tremor result when neurons are deprived of fuel. Severe hypoglycemia can lead to unconsciousness and death. The lipid-soluble hormones, steroids, and thyroid hormone bind to receptors typically located inside cells. Most of these intracellular receptors are located in the nucleus. Each receptor has two binding sites, One site binds the hormone, one site binds to DNA. In the absence of hormone, the receptor is bound to a chaperone protein that masks the DNA binding site and probably also acts to stabilize the protein. The receptors are not active until a hormone binds to them. Drag the hormone to the binding site on the receptor to observe its activity. A hormone binds to a receptor, the receptor changes shape. This activates the receptor and causes the chaperone to disassociate from it. If it is not already in the nucleus, the hormone receptor complex enters the nucleus. Hormone receptor complexes act as transcription factors. They bind to specific regions of DNA called hormone response elements. Typically, the binding results in activation of or turning on a particular gene. When a gene is activated, messenger RNA, mRNA, is produced and enters the cytosol where it is transcribed to form proteins. Typically, the proteins that are synthesized are enzymes that catalyze biochemical reactions and cause the response of the cell. The properties of transcription factors are far more complex than the description above. Transcription factors can regulate more than one gene, and they can also decrease or turn off the activity of genes. Transcription factors for different hormones can bind to the same hormone response elements, although each transcription factor activates a specific group of genes. To view the animation again, drag the hormone to the receptor. Click Continue to proceed. On this page, we will study the mechanisms used by cortisol and thyroid hormone to induce cellular responses. The action of cortisol follows the typical pattern for steroid hormones. It binds to glucocorticoid receptors that are present in nearly all cells. Some of the many cellular responses to cortisol are listed. Cortisol acts to maintain high levels of fuel molecules, maintain blood pressure, and modulate body defenses, all necessary for the stress response. We will study the stress response in Topic 5. The thyroid hormones, T3 and T4, enter cells by diffusion, and to some extent by carrier-mediated transport. Once inside, T4 is converted to T3, the active form of the hormone. Receptors for thyroid hormones differ from steroid hormone receptors. They are bound to the response elements of the DNA. T3 binds to the receptor, causing gene activation or eliminating the inhibition that was present, thus allowing genes to be expressed. Small amounts of T4 may also bind to the receptors. Receptors for thyroid hormones are also found in the cytosol and mitochondria, but their function is poorly understood. Nearly all body cells have thyroid hormone receptors and require thyroid hormone for normal function. Some of the many effects of thyroid hormone are listed. Thyroid hormone is the main regulator of metabolic rate in the body. Here's a summary of what we've covered. Hormone receptors are proteins located in the plasma membrane or the interior of the cell. Water-soluble hormones bind to plasma membrane receptors, and lipid-soluble hormones typically bind to intracellular receptors. Receptor activation engages cellular machinery that leads to the response of the cell. The effects of a single molecule of hormone are amplified by the cellular machinery. Insulin deficiency or insensitivity causes diabetes mellitus, whereas hypersecretion causes hypoglycemia. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.